Hello, dear students. I welcome you all on behalf of Sri Jayendra Range Zone classes. Dear students, today we are going to start with a new chapter that is anatomy of flowering. A new term, flowering plant. Flowering plant and the human, they are the angiosperms. But here today, the another thing which we are going to discuss, that is anatomy of flowering plant. Now, when we talk about anatomy, it is going to talk about the internal structures. It is going to talk about what? Internal structure. So this anatomy, it is nothing but a branch of botany which is going to deal with the internal structure and organization of plant and plant organ, or we can say the plant parts. So it is overall study of internal structure of the plant. Now, it's study, if we talk initially in detail, done by Nehemiah Group, that is shortly known as N Group, and who is considered as father of plant anatomy. Okay, so you should remember this. The study of internal structure is anatomy, and father of plant anatomy is Nehemiah Group. In this chapter, we mainly talk about the tissues, the types of tissues which are going to play there, what are the functions associated with, what are the, we can say, how these tissues are arranged, okay, how that grow, and in that how the growth, girth wise growth occur, that all, and what is the internal arrangement of tissues in dicot, monocot, and in dicot, monocot, root stem, in root stem and leaf, that we discuss here in detail. Okay, so I hope you understood this, what is anatomy. Now, if we discuss further, what is histology? First we discuss, because we are going to discuss mainly about the tissues in this. So the study of different tissues in the plant body is comes under one branch of biology, that is histology. So it is going to deal with the structure of the tissue or different tissues. So this is just anatomy of dicot root. We will discuss in detail what this all things are saying. But if we observe this at this microscopic level, the structural organization of different plant parts, then it is a part of anatomy. So the study of internal structure, that is the arrangement of various tissue of a plant, is called anatomy. So anatomy mainly deals with the arrangement or the study of internal tissues present within the plant. Now, if we talk about the initial part, we can say that the structural similarities and variation in the external morphology of larger plant. So we easily see this similarities or variation in the external morphology of larger plant by studying their external structures like root, stem, leaf, and their modification, as we have discussed in the previous chapter. We can easily study this for the larger plants like the angiosperm and gymnosperms. But if we are to study we have to study the internal structure, or one also find so several similarities as well as differences. But to study these internal structures, there is somewhat difficulty. Okay, and you can see if internal structure we go within cell, what are the different organelles are present, how they are arranged, that we can see. Or you can see, you will see the leaf anatomy, this complex leaf anatomy is visible. So if we want to study these things, then it will be somewhat difficult. So if we go on this internal organization, internal structure and functional organization of higher plant, that comes under the part of this anatomy. So the basis of unit of plant is cell. You know that. We know that cell combine or organize and form a tissue. So tissue is nothing but a group of cells. And when tissue further organize, they are going to form organ. So cells are going to form the tissue and tissue are going to form the organ. So this way the organization is going on and different organ will interact and that will be a formation of whole, we can say a plant body. In plants, if we talk about different organs like root, stem, leaf, flower, fruit, all this show differences in their internal structure. They show differences in their internal structure, and mainly we are focused on that part. Then we can justify the study of this chapter. So initially, one scientist was there whose name is Catherine Esau, who is the plant anatomist. Okay, who started mainly study with this anatomy in detail initially. She was born in Ukraine in 1898, and she reported that curly top virus, it is a one type of virus in plant, spread through or via through a plant via food conducting or phloem tissue. Means this curly top virus particularly spread through the phloem tissue or conducting tissue. And the anatomy of seed plants means the gymnosperm and angiosperm by Catherine Esau as a one book published in 1960. So this is the book. So she was elected to the National Academy of Science. In National Academy of Science also she was elected, which is one of the prestigious institute, and she received the National Medal for of Science from President George Bush in 1989. So this is the what history of this Catherine Sew, who is the one of the important known scientist of that time. So study of different tissue in plant body is the anatomy, as we have discussed. Study of tissue, different tissues, it is histology. And when we talk about arrangement or organization, that is, they are going to talk about the anatomy. Study of internal structure or arrangement of various tissues of the plant, it is anatomy. So see, remember, what is the difference between histology and anatomy? Histology is study of different tissues, just. And when we talk about arrangement or when we talk about internal structures of plant, then we talk about the anatomy. And the work considered as a Webster dictionary of plant biology is okay. So this the anatomy of seed plant, which was written by Catherine Sau, it was referred to as a Webster's of plant anatomy in an encyclopedia. Okay, so work considered as Webster's dictionary of plant anatomy by that is the nothing but work of this Catherine Zhao. It is given in the just initial part NCRT page of NCRT. You should remember that. Catherine Asau reported that the curly top virus spread through a phloem tissue. Just we have discussed. So answer is going to be a phloem tissue. So now after this tissue, which we are going to discuss here mainly the meristematic tissues. To say here meristematic tissue are then permanent tissue. First we discuss the type of tissue. Okay, and first we talk about the mainly the tissues. Okay, so what is anatomy? Who people are involved in this? What they did that we have discussed. What are the branches associated with that we have discussed. Now we are going to talk about the anatomy. That we are going to talk about the arrangement of these different organs and arrangement of tissue in different organs or different arrangement of different tissues. And so to study that, first we need to understand the tissue. So what is the tissue? A tissue is a group of cells. Yes, we have studied that group cells will form a tissue and tissue is going to form an organ. But the cells which are going to form a tissue, they should have a common origin and perform common function. Okay. So tissue is nothing but a group of cells. They are having a common origin and usually they perform a common function. Structure may vary, then it is called as tissue. And as we study the internal structure of a plant, we come to know that a plant is made up of different type of tissues. And based on the cell being formed are capable of dividing or not, tissues are classified into two groups. Now, in a tissues, the cells which are comprises and forming this tissue, they are of two types. Either they are having a capability of division or they have lost this capacity of division. If they are in a state of division, they are known as meristematic tissue. If they are not in a state of division, it is known as permanent tissue. Or we can say the tissue, meristematic tissue have a cells that are meristematic cells which have a capacity of division. They are in active state of division. But permanent tissues have a cells which have lost their capacity of division. So first we talk about meristematic cells and then this particular. But we remember this tissue was classified into two groups by Carl Nageli. Tissue classified into two groups by Carl Nageli. And he divided this tissue into the two type meristematic tissue or permanent tissue on the basis of capability of division. On the basis of capability of division. And in this tissue, there are two tissues are there, meristematic tissue and permanent tissue. If you talk about meristematic tissue having a dividing cells, it is a group of dividing, so it is undifferentiated tissue. Whereas permanent tissues have a cells which have lost their capacity of division and gain a particular function. So it is a group of fully differentiated cells. Okay. And this meristematic tissue, if we talk, they are composed of immature dividing cells. So dividing cells are considered as immature, meristematic cells are considered as immature. But permanent tissue composed of mature non-dividing cells. And if we talk about tissue, then the term tissue was coined by was coined by Nehemiah group. Okay, that is the thing which is done by the Nehemiah group. So it is a division classification. Now, first we talk about the meristematic tissue. In a plant, the specialized region of active cell division are called as meristems. Means meristems are the nothing but a region where this meristematic tissue are located. Are the site where the meristematic tissue are located. So in Greek word, meristos means divided. Because of this, the term or a region where meristematic tissue or dividing tissue are located, that region is named as meristems. Usually, if we talk, plant have different kind of meristem. In this, we are going to discuss the apical meristem, intercalary meristem, and lateral meristem. That we are going to discuss. First, we are going to discuss the apical meristem. And this meristem, as I told you, they are present at the different site in the particularly plant. The apical meristem, if we talk, is the meristem which occur at the tip of root and shoot and produce primary tissues. The meristem which is located at the tip of root or the tip of shoot, and it is going to give rise to the primary permanent tissue, means parenchyma, parenchyma, and sclerenchyma. Then this meristematic tissue are known as apical meristem. These are divided based on the position. We remember apical meristem, intercalary, and lateral meristem are divided based on the position. So the position is apex. Because of this, it is known as apical meristem. See, this is shoot meristem, shoot apical meristem. Actually, we can call it shoot apical meristem, and this is a root meristem, which is also known as root apical meristem. So the apical meristem, which is present at the tip of the shoot, it is known as shoot apical meristem, or you can call it a SEM. And this root apical meristem, also known as root meristem, which is present at the apex of root. This root apical meristem helps to grow in a length of root, as well as shoot apical meristem also help the plant to grow in a length as well as add the particular primary permanent tissue. Now, root apical meristem occupy the tip of the root, as we have discussed. The shoot apical meristem occupy the distance mode region of the stem axis. So this is the distance mode region, which is a shoot apical meristem, and this is the basal most region, which is a root apical meristem. During the formation of leaves and elongation of stem, when stem elongate and new leaves are going to form, some cells, means apical meristem is going and it is forming a new leaves. It is helping in the elongation of stem. But at the same time, some cells are left behind from the shoot apical meristem. And the cells which are left behind from the shoot apical meristem, they constitute the axillary bud. So such buds, axillary buds, are present in the axilla of leaves. So the question is, what is axilla? Obvious. And this axillary bud, if we talk, they are capable of forming a branch or a flower. See, if this is the stem, this is the growing part, this is the node, and this is the leaf. The angle which is formed here, this angle is known as axil. So axil can be defined as the angle between petiole of leaf and younger stem. This is the younger stem, growing stem, and this is the petiole of leaf. So the angle, this is known as axil. And whenever any bud is present in this axil, this bud is known as axillary bud. Now these are the structures which are showing the particular internal arrangement of apical meristem of root and shoot. If you can see root, the basal portion, in this you will see the root cap. Initials of root cap are here. Okay. Then there is a root cap. These are the initial of root cap. This is the root cap. And initial of central cylindral cortex. Central cylinder is formed by here, but here you will see this location, and this is representing what? A root apical meristem. So on the below side, the root apical meristem is covered by some of the initials of the root cap as well as root cap. And above to this root apical meristem, you will see the presence of cortex, central cylinder, all tissues you can see. Okay. So this labeling is important. Sometimes you will get a question from this part, and they will ask that what this particular labeling represents. Then you cortex, this root apical meristem, this protoderm, this all you should know. And on the other side, in the shoot, if we talk, which is the distal moat region on the aerial part or the axis of the stem, you will see here the presence of this particular shoot apical meristematic zone. Shoot apical meristematic zone means it is nothing but shoot apical meristem. Now, here you can see this leaf primordia. Leaf primordia is nothing but initial leaf which are going to develop as a leaf. And here you will see this particular axillary bud. So axillary bud is what? Nothing but the some cells are left behind from the apical meristem, and they are accumulating and forming this axillary bud, which is able to give rise to the new branch or a leaf, or new branch or a flower. But this tissue, if you talk, this one, this is, they are calling a differentiating vascular tissue. Vascular tissue means xylem and phloem, which are forming. Obviously they are going to be the primary xylem and primary phloem which are forming here now. So that are forming, that are differentiating from the dividing cells, okay, in the initial part. So this labeling is much important. Sometimes they ask from this part, and you will get an option like undifferentiating vascular tissue, differentiating vascular tissue, such type of words you can see. Okay, so that is the important. Now here it is important to understand this term like undifferentiated, de-differentiated, differentiated. What are they that we need to understand? We will understand in the coming slides. So this is the actual diagram of the, we can say, leaves, apical meristem and the root apical, shoot apical meristem and the root apical meristem. In the root apical meristem, if we talk, you will see the central cylinder, you will see the cortex, you will see the protoderm, you will see the root apical meristem, and you will see the root cap. What are the labeling we have discussed in the NCRT diagram? Actually, they look like this. Difficult to differentiate. Same way, if you talk about shoot apical meristem, they have leaf primordium, initial leaves, they have shoot apical meristem, they have meristematic zone, shoot apex meristematic zone, then they have a axillary bud and differentiating vascular tissue. Okay, so such particular labeling are given in NCRT. That NCRT diagram you should remember. Usually they ask the question from that diagram directly, so you should remember that. Now, leaf primordia, what is? So it is a nothing but a group of cells which develop into a leaf. So I called it initial leaf. Seen as a small bulges just above, just below the shoot apex. Means this is the shoot apex, and from the just below shoot apex, this are getting originated. Okay, so they are the initial leaf. Now another type of meristem which are seen, that is known as intercalary meristem. Now this apical meristem which we have studied, that usually example of a primary meristem. We will discuss what are primary meristem. Okay, and they are this apical meristem which we have studied that are actually responsible for the increase in the length of plant organ. Means they are responsible for the primary growth. Okay, the lengthwise growth is considered as a primary growth. Intercalary meristem, if we talk, this is the meristem that occur between the mature tissues. It is known as intercalary meristem. Where it is located? Between mature tissues, means between the permanent tissues which have already stopped their capacity of division. Okay, there intercalary, calary means in between. The particularly intercalary means inter between the permanent tissue or mature tissue it is located. So this is the intercalary meristem, as you can see. They occur in grasses and regenerate the plant part by the grazing herbivores. One of the important points. Suppose it is actually present behind the apical meristem, not at the apex. They are intercalary, present below or behind the apical meristem and between the mature tissue. So if some grazing herbivores eat the apex of leaves, especially grasses, but in grasses the regeneration is observed. Why? Because the some meristem which are located between the permanent tissue or mature tissue like intercalary meristem help to regenerate the plant part removed by the grazing herbivores. Both apical meristem and this intercalary meristem are primary meristem. Actually, both are primary meristem. Okay, means they are helping the primary growth. They are helping in the lengthwise growth. Because they appear early in plant life, they are known as what? Primary tissue. They contribute for the formation of the primary plant body. Okay, we will come to the secondary one, but this is the what? A particularly intercalary meristem. So you can mark it here that this is the apical meristem is present here, but this is the particularly present between the mature tissue, so it is a intercalary meristem. Now the question is where this intercalary meristem are present. So if we talk about location, then we come to know that they are present at the base of at the base of internode of internode of monocot stem. example, grasses, bamboo, sugarcane, etc. Also present at the base of leaves. And by the activity of this meristem, which is present at the base of leaves, the length of leaves getting increased. So this intercalary meristem also help to increase the length of the plant organs. These both apical and intercalary meristem are actually primary meristem, and which occur initially, which originate initially, but the meristem that occur in mature regions of the root and shoot of many plants, but appear later than the primary meristem. They appear later in the life. They originate later in the life. Then they are known as secondary or lateral meristem. You can see lateral meristem are marked here. This lateral meristem, particularly those that produce a woody axis, means this lateral meristem are helpful in the increase in the girth, increase in the thickness of the plant organ. Because of this, they are present sidewise. You can see its location where they are present. They are present sidewise. Why they are present sidewise? Because they help in the girth wise growth or thickness wise growth of a plant organ. So they are cylindrical meristem. They are also known as cylindrical meristem because they are present laterally and cylindrical in shape. They are responsible for producing the secondary tissues. They are responsible for the secondary tissues, means they are helping in the secondary growth. When secondary growth occurs, the secondary tissues means the xylem and secondary phloem are getting produced. This secondary xylem and secondary phloem are produced by the activity of this lateral meristems. Other secondary tissues are also present that we will discuss. Okay, so these are the lateral meristems. Now, if we talk about the examples of this lateral meristem which occur later in life, so one point we should write here that lateral meristem help in girth wise or thickness wise growth. So increase in girth or increase in circumference or thickness of a particular axis, it is caused by this lateral meristem. And if we talk about the examples of lateral meristem, the vascular cambium, interfascicular cambium, and core cambium are the lateral meristem. Actually, this vascular cambium is nothing but intrafascicular vascular cambium. It is intrafascicular vascular cambium. Intrafascicular vascular cambium is present within vascular bundle. So this interfascicular vascular cambium, if you talk, so this intravascular vascular cambium are present within, we can say, vascular tissue. Within vascular bundle. So these are present within vascular bundle. Within vascular bundle means what? We can say they are present. See, suppose this is the xylem. This is the primary We can say this is the primary phloem actually. This is primary phloem, and this one is primary xylem, and this is the whole cross section of this part. So this is primary xylem, this is primary phloem, and the vascular bundle which is present within this, we one xylem and one phloem form one vascular bundle. So the cambium which is present within vascular bundle, it is known as primary xylem. This is known as intravascular vascular cambium. But this is one vascular bundle, this is another vascular bundle, this is another vascular bundle, suppose, and this is the one vascular bundle. The tissue which is, or a vascular cambium which is present between the vascular bundle, it is known as interfascicular vascular cambium. It is present between vascular bundle. Okay, and when they are present between vascular bundle, then they are known as interfascicular vascular cambium. And apart from this, you will find one more vascular cambium which is present on the outer side of this vascular cambium, that is known as cork cambium. So primary and secondary meristem, if we talk, they form a new cells, and this newly formed cells become structurally and functionally specialized. They get specialized, or they gain a particular function structurally and functionally. And when they get particular function and structurally get modified or specialized and develop into the permanent tissue. Okay, so we will discuss what type of permanent tissue are found by this primary and secondary meristem. But these three are the what? A lateral meristem, which help in secondary growth. Okay, so this is all about the what we can say primary and sorry, we can say what particularly apical meristem, intercalary meristem, and the lateral meristem. This all division of meristem into the apical, intercalary, and lateral meristem is based on the location or position in the plant body. But this meristem in plant are also divided based on the origin and development. So classification of meristem based on origin and development. Okay, based on origin and development, we are going to classify them. And based on origin and development, they are classified into three types. First type is known as promeristem, or we can say primordial meristem, or meristem. These all are the specifically we can say promeristem. Now this promeristem develop during or develop during embryonic stage, and this promeristem form primary meristem, that form primary meristem. So if we talk about the example of this promeristem, then it is nothing but a embryonic meristem. Then it is embryonic meristem. After this, the second type is primary meristem. Now second is the primary meristem. So a primary meristem, if we talk, then primary meristem developed from promeristem. It actually developed from promeristem, and it is known as primary meristem. Now this primary meristem actually appear early in life of plant or originate early in life of plant, and they contribute they contribute to the formation of primary plant body. So primary plant body is actually formed by this primary meristem, and these cells are always in a division phase. Cells are always in a division phase and form primary permanent tissue and form primary permanent tissue like parenchyma, collenchyma, sclerenchyma. So if we talk about example of primary meristem, then they are nothing but, as we have discussed, apical meristem, intercalary meristem, and intrafascicular vascular cambium. This intrafascicular vascular cambium is also known as fascicular vascular cambium, which is present within the vascular bundle. And the apical and intercalary meristem also originate initially early in the life, and in this also intrafascicular vascular cambium also appear early in life, so it is known as primary meristem. But see, by location wise, this intrafascicular is lateral in position, but origin wise it is primary meristem. After this, if we talk about the secondary meristem, then the secondary meristem developed from primary permanent tissue. Developed from primary permanent tissue. Now, primary permanent tissue are nothing but parenchyma, collenchyma, or sclerenchyma. So they are in a non-dividing stage. Means secondary meristem is developing from the non-dividing cells, and primary meristem was initially developing from starting. It was in a division phase, but the cells which was non-dividing, they get converted into the dividing. Then they are known as what? The secondary meristem, and they are developing from the primary permanent tissue by de-differentiation. So this secondary meristem appear later than primary. This appear later than primary meristem. And if we talk by the activity of secondary meristem, if we talk about this activity of secondary meristem, by activity of this secondary growth or girth wise growth, take place. And if we talk about example of this secondary meristem, then it is interfascicular vascular cambium and cork cambium. Okay, these are particularly. So what is a differentiation, de-differentiation, and redifferentiation that we need to discuss? Then it will be so. Initially, from early life, which the T meristem which are present are the primary, like apical, intercalary, and intravesicular. But secondary meristem appear later in life from the non-dividing cells. So if we talk about, suppose this is a dividing cell, and this is non-dividing cell. So dividing cell, if we talk, they are in active state of division, means they are undifferentiated tissue. Now, when dividing cell convert into non-dividing cell or it loses its capacity of division, then it is known as differentiation. So the cells which are formed by the differentiation are they have lost their capacity of division and they have structure functionally specialized and they gain a particular function. Now they are in a non-division state, they are in non-dividing state, but somehow if plant need this in a division state again, then this non-dividing cells start dividing again, then it is known as differentiation, where non-dividing cell convert into the dividing cell. And this dividing cell, which have formed after the de-differentiation, that if again somehow lost the capacity of division, then they are known as redifferentiation. So this differentiation produce differentiated cells, and differentiated cells are non-dividing, whereas de-differentiation produce de-differentiated cell, and this de-differentiated cell are dividing cell, where the redifferentiated form are redifferentiated cells, and this redifferentiated cell are non-dividing cells. Okay, so this way the all particular first. So the primary meristem and promeristem are the undifferentiated tissue, but primary permanent tissue like intercalary, parenchyma, collenchyma, sclerenchyma are formed by the differentiation. So primary permanent tissue are the what? Particularly we can say they are the differentiated tissue. But secondary meristem is a dedifferentiated tissue, and the secondary permanent tissues, secondary xylem, secondary phloem, are the redifferentiated tissue. And you will understand this in the later part in more detail as we go on studying. Okay, so I hope you understood this. Now, after this division, one more thing we have to note down here with respect to this, that is the one scientist were named whose name is Haberland, and this Haberland divided eumeristem, divided eumeristem. This eumeristem is nothing but we can say a primary meristem or apical meristem. Primary meristem into three regions, into three regions on the basis of function. On the basis of function, they are divided into three regions. And these three regions are, first one is the protoderm. Protoderm, if we talk, it is the outermost layer of eumeristem, and this particularly outermost layer of the eumeristem form epidermal tissue system. They form epidermal tissue system, which include epidermis, stomata, stem hairs, root hairs, and all this. So if we talk about the meristem like this is protoderm. This is procambium. So second is procambium. It forms vascular tissue system, and this vascular tissue system consists of xylem and phloem, that is vascular bundle. Whereas third is ground meristem. And this ground meristem form ground tissue. In ground tissue are include what? Cortex, pericycle, teeth, medullary rays, all things are produced by this particular ground meristem. So this way, these three are located which are going to form this particular one, as Haberland divided this three regions based on the function. Okay. So this is the all about what particularly we can say meristematic tissue. So tissue are mainly classified into two groups, that is meristematic tissue and the permanent tissue. The meristem that occur at the tip of the root and shoot to produce primary tissue are known as apical meristems, and they are root apical meristem and shoot apical meristem. Apical meristems are, if we talk about apical meristem, they are always primary. Shoot apical meristem is present as stem axis, obviously, distal small region of the stem axis. The meristem which occur between the mature tissue is known as intercalary meristem tissue. Intercalary meristem. And this is the only type of a primary meristem. Okay, so here we are at the end of our meristematic tissue, and we have completed meristematic tissue in detail. Now in the next lecture we will be discussing permanent tissue in detail. Okay, so I hope you understood it very well manner. Thank you.